Oh. Okay. Okay. It's interesting, actually. Oligo peptide and full blown proteins. Okay. All right. That's pretty simple. Fatty acid origin. So I was sick. So I feel like the crazy thing is I spent like 2 or 3 days off, and I feel like the whole world is crashing down on me. Hmm. Release from. Okay. Mechanisms of hydrophilic signal molecule action. Hydrophilic yeah, short half-life. Right. Since the signal molecule first messenger does not enter the cell, the receptor ligand complex causes a second messenger to be produced or released within the cell. Okay, so can't diffuse cross membrane, so needs secondary messenger to released, produced as a result of ligand receptor complex. All right. The second messenger acts as a coenzyme cofactor as coenzyme cofactor in regulation cell enzymes. Okay. So we have oh, okay, this is a good picture actually. So we have a membrane, we have a thing, we have a signal. This is S. This is R for receptor. This causes a thing, which is the secondary. Then we have an inactive thing, which then via this becomes active. And then that active thing then causes and then now I don't know what all this is. Okay. All right, now intracellular receptors for lipophilic signal molecules. Let's see what's going on here. So in the cytoplasm, this is the nucleus, DNA. Okay, so this is the cytoplasm. This is the blood. The carrier protein, it releases, this thing diffuses. We have a receptor protein. What is that? I don't know what that is. The receptor protein, oh, this is the transcription factor or something? Okay. Okay, so I guess what I'll just say of this is the lipophilic So what's happening here though? This is only modulates enzyme activity. So let's just ask that most of the hydrophilic signaling molecules in the body do not modulate gene, do not participate as gene regulators, but mainly act on already produced proteins. Whereas the lipophilic are more are the gene regulation signals. Kinda, but not as clean. Okay. Yeah, okay. Yeah. It's broadly true that most hydrophilic, so the hydrophilic. All right, okay. Right, yeah. So, and secondarily affects gene reg. Yeah. Organs specialized for secretion. Does crine mean blood? I don't know. Endocrine glands. So wait, hold on. Connecting cells disappear. DIVA cells remain discrete into capillaries. Okay, yeah. So I'll write that. So the exocrine glands have a duct. I'll say outward, maybe. I don't know. And some glands are exo and endo, like the pancreas. Pancreas secrete digestive enzymes into small intestine via bile duct, and insulin glucagon via capillaries. Okay, good. Alpha gluc, beta insulin, and gamma or delta somatostatin. Which I don't know what somatostatin is. Interesting word though. Somatostatin. Gonads also. I'm going to make a new word, ambicrine. Ambi means both, I think. No, is it amph? Oh, amph. Alright, I'm going to say ambicrine. Ambi means both, so... gametes through duct and sex steroids via blood. All right. What is this? This is 12. So we have neuroendocrine glands. Was that up above here? In not really. Was there a thing on the type of glands? I don't think so. All right. So I guess the main thing is that neurohormone goes to blood, but a neurotransmitter specifically goes to synaptic cleft. Okay. Some endocrine glands have both neural and epithelial components. Ooh, I never actually noticed that. Adrenal? I thought it was one thing. Adrenal. A medulla and a cortex. Okay, some endoglands are neural and epithelial. Adrenal gland. So the adrenal medulla is neural. The cortex is epithelial. So that means that I guess you can stimulate secretion neurally, but then you can also, I don't know what that means really. Right, here we go. The adrenal gland, modified sympathetic postsynaptic neurons. Let's try to look at the diagram first. The spinal cord nerve signal. What is this? The brain? I don't know what this is. So nerve cell to the inside of the adrenal, into the adrenal medulla. Epinephrine and norepinephrine. Okay, and then the adrenal cortex. Okay, and then fasciculata and reticularis. Yes, reticularis. So we have glomerulosa, fasciculata, reticularis. All right. Three subregions. Mineralocorticoids. So the mineralocorticoids. This is like aldosterone. I forgot the function of aldosterone. And then we have glucocorticoids, which are like cortisol. And then finally, sex steroids. And this is going to be like androstenedione. Androstenedione. This might be a good androstenedione. And then I don't know if we need corticoid. I feel like corticosteroid is pretty cortex, and then I guess like cholesterol derived or something. I don't know. Whoa. Not all hormones are secreted by specialized glands. Regulation of blood glucose by antagonistic negative feedback loops. Okay, this should be pretty basic. Pancreas regulates blood sugar levels. Glucose is taken up or released by liver as glycogen stores. Glucagon from alpha cells stimulate the liver to turn glycogen into glucose. This increases blood glucose. Then insulin from beta cells stimulates the liver to turn glucose into glycogen, so it goes down. Okay, that's a pretty simple thing here. I feel like this is also something I know. Okay. Insects and others also use antagonistic hormones. Ecdysone induces... Oh, interesting. In insects, ecdysone induces new cuticle. Yeah, because it literally says molting hormone. Ecdy means, yeah. And then juvenile inhibits terminal molt. All right. prothoracic gland. Prothoracic gland. All right, pituitary gland, the master gland. Oh, we're getting into a lot of this Latin. This all looks Latin. Adenohypophysis. Whoa. I don't understand. The anterior lobe is pointing to... Okay, this is the anterior lobe, and this is the posterior lobe. Okay, fine. Okay, I see. Pituitary is the master gland. Which, it's under the hypothalamus. Divided into the posterior anterior lobes. The post is from hypothal neural tissue, but anterior is an epithelial gland from the roof of the mouth. Wow, roof of mouth. I was not expecting it to say that. That's weird. Where's the hypothalamus? Okay, so okay, so I guess we need these words. Okay, adenohypophysis. Adenohypophysis. And then neurohypophysis. I guess the thing that I want to know is, what does adeno mean? Adeno means gland in Greek. Oh, really? Wow. Interesting. Holy shit. Does he... If he's putting this picture here, I guess he wants us to... Supraoptic? I've never seen supra. Paraventicular. So around the ventricle? Well, there's that. I guess, yeah, I guess that's what I mean. But no, that doesn't make any sense. What? Maybe it means chamber. I think it just means chamber. I don't think it. Yeah, any cavity. It's not specific to the heart. So around a cavity nucleus, I guess. Hypothalamus infundibulum. Infundibulum. Hypothalamo hypophyseal tract. Hypophyseal. I don't know what physeal is. ADH. Okay, let's just figure out this chain of logic. Yes, so ADH causes the aquaporins to form in the collecting duct of the nephron. So therefore, the kidney is going to reabsorb more water, so the blood osmolarity is going to go down, and the blood volume is going to go up, and blood pressure goes up. Okay, good. Oxytocin what? Okay, I was just going to write that the pituitary produces ADH and oxytocin. This is specifically in the posterior, I guess. Okay, so we have a positive feedback. Oh yeah, we've seen this before. Oxytocin stimulates the uterus to contract, which stimulates even more oxytocin to. Oh, here we go again with this prostaglandin. Prostaglandin. If it's on the slides twice, you can pretty much assume that he wants you to know it. Stimulate more contractions of uterus. So you actually have two feedback loops. You have this feedback loop and then this feedback loop. All right, here we go. Anterior. What does trophic mean? Heterotroph. Doesn't troph mean eating? Oh yeah. All right. Holy cow! Look at this diagram, man. All right, here we got the paraventricular nucleus, the supraoptic nucleus. So it looks like there's some capillaries here. Okay, this is the mammary gland. Okay, it's stimulating something. I guess it's releasing something called prolactin. It's releasing something called TSH, ACTH in the adrenal. I think this is called luteinizing hormone. I think I remember reading about this. Okay, and a bunch of gonadotropins and growth hormone. Okay, the anterior pituitary master gland. Stimulates activity. Oh, it's like the precursor activity of other glands. I see. Okay. And stimulates growth in target tissues. Cool. All right, the pituitary. This is why you hear all these freaking people talk about the pituitary like it's... Because it is the precursor gland. It's the preeminent... I don't know what... Preeminent? That actually seems like a good word. Emanate. Preeminate. Why is it the preeminent? Why isn't it just the eminent gland? Preeminent. Preeminent. Oh, but eminent does not mean emanate, right? Emanate. Emanate. As from a source. Eminent and emanate are different words. Eminent and emanate. Emanate are different. Preeminent, eminence, your eminence, or whatever, I think. But emanate, all right, I got it. So it is the preeminent gland. It's the superior gland. Releasing, stimulating. This slide stinks here. Can't read it at all. Once we read the textbook, I'll get that. Feedback control of the anterior pituitary. For example, sex hormones. The hypothalamus secretes releasing hormone to stimulate the anterior pituitary. Oh, here we go. Gonadotropin releasing hormone. The anterior pituitary then secretes gonadotropins, luteinizing hormone, and that other one, to stimulate gonads to grow and mature, and 2, to secrete sex steroids. Dude, I wonder what would happen if you just took like a bunch of that. Like, you'd probably freak out. Sex steroids from gonads. Feedback. To inhibit the anterior pituitary from secreting more gonadotropin. Okay. But also, it inhibits the hypothalamus. Okay. So there's a double thing. All right. Oh shit. They're grapes turned to raisins. Ew, bro. What the hell? If a bodybuilder takes anabolic steroids, the steroids act like other androgens and inhibit the pituitary from secreting gonadotropins. Thyroid. What does thyroid mean? The thyroid is an endocrine gland in the neck. It secretes hormones that influence metabolism. Oh. Okay. All right. Feedback control of the interior pituitary even more. Okay. So all right. So it sounds like understanding this is going to be important. Hypothalamus secretes releasing hormone. I see. Thyrotropin releasing hormone to stimulate the interior pituitary. Then the interior pituitary secretes thyroid stimulating hormone. Okay. Then the thyroid does something to inhibit this, but then also causes the growth of the thyroid. Okay. The interior pituitary secretes thyrotropin to stimulate thyroid to grow and secrete. Okay. Tyrosine from thyroid feedback to inhibit pituitary from secreting more TSH. But iodine is needed to synthesize tyrosine. If the diet is deficient in iodine, thyroid cannot make thyroxine. That's actually a better way to say it. Thyroxine. If no thyroxine, no negative feedback on interior pituitary. Interior pituitary continues to secrete. Okay. A goiter. What the hell is that? All right. Pars intermedia. In most vertebrates, a portion of the pituitary anterior lobe adjacent to the posterior lobe develops into an intermediate lobe. Principal activity is secreting melanocyte stimulating hormone. MSH promotes hyperpigmentation from increased melanin production in melanocytes in skin and hair. Cryptic coloration, sexual territorial advertising. Okay. Pars intermedia. Alternate processing of the same prohormone polypeptide pro-opiomelanocortin. Whoa. What a name. In the anterior pituitary, POMC cleaved to form endorphins. Adreno is adrenal. Cortico is cortex. So the adrenocorticotropic hormone. in the intermediate lobe. Bro, why are we learning this? This is crazy. ACTH fragment is further cleaved to form alpha MSH. In humans, intermediate lobe is greatly reduced but present. Syndromes that cause an overproduction of ACTH also result in elevated MSH and hyperpigmentation. Alright, this slide I didn't get at all. This pars intermedia. I guess the pituitary is pretty important. Alright, endocrine pathologies. Abnormal hormone titer. Primary pathology. Cause the endocrine gland secreting that hormone. Wait, I feel like that... What? Caused by a factor. Regulated. What? Hypersecretion of hormone. Idiopathic? What does that mean? Gland. Turned on for no obvious reason. Tumor hyperplasia. Secondary hypersecretion of trophic hormone. Hyposecretion. Enzyme defect in biosynthesis of the hormone. Autoimmune destruction of secretory cells. Receptor defect insufficiency responding to trophic hormone. What is trophic hormone? He probably said it when he gave this... I guess when they say trophic, they mean like it's feeding another gland. So like if you say thyrotropin, the thyroid is the one feeding, I guess. I don't know. Kind of makes sense. Abnormal hormone response. Number of receptors in target organ desensitization. Mutant defect receptors. Okay, understanding this freaking endocrine pathology is probably important. All right, here we go. Let's get to the good stuff. Electrochemical communication. Yeah, the membrane potential, excitability, conduction, transmission. All right. So these are kind of the aspects of electrochemical communication. So we have neurons, and we have the membrane potential. We have excitability. We have conduction and transmission. Neurons conduct electrochemical impulses and transmit messages to other cells. Actually, you know what would be good to look at too is oh, here here we go. Oh, God. We get a lot of words. Let's see. What do we have here? A lot of these are just going to be like anatomical things. It's not going to be like his aorta, appendicular skeleton, archenteron, atrium, axial skeleton, blastopore, blastation, bone. Oh, I don't know about that. Carina procoracoid for Kula. Oh, I don't know any of that. That's crazy. Carpometacarpus? What the fuck? Cephalization, cephalocordata, chondrichthes, chondrichthes. I think cond is like cartilage, but I don't know that one yet. Chordata, ciliated tissue, circulatory system, clasper, don't know that. Cleavage, I know that basically because miro means partial and hollow means complete. Okay, cleavage, radial, spiral, determinate, indeterminate, yeah. I always forget which one is which. I'm pretty sure that we are radial, which is deuterostome. Cloaca, coelom, connective tissue, cranium. Ooh, I don't know any of that. Crop, deuterostome, diaphragm, differentiation, digestion, dorsal nerve tube, ectoderm, gelatinous shelled egg, embryo, endoderm, endoskeleton, exoskeleton, epidermis, epithelial tissue, esophagus, eucoelum, extracellular matrix, extra embryonic membranes. Oh, yeah, we're going to need to know this. Allantois, amnion, chorion, yolk sac. Yeah, yeah, okay. I need to study that. Facial bones, whoa. Zygomatic arch, rostrum, hard palate. Fins, paired, unpaired, pectoral, pelvic, dorsal, anal, caudal. Okay, yeah, that's chill. Fish, fishes. Oh, okay. Fish form. Oh, yeah, we're going to need to know this. And should I just add these right now? Nah. Okay, we'll do this in a minute, though. I really do want to finish out the Anki. I haven't done Anki in a month. Flagellated cell. All right, I'll be back in a second. Gastrula, gastrulation, genitalia, gills, gill arches, gill filaments, gill rakers. The rakers, I think, redirect the food, right? Gizzard, gnathostomes. I don't know what the gnathostomes are. Gonad, heart, hemocele, hemolymph, hermaphrodite, heterodont. Oh, wow, we're going to need to know teeth. All right, yeah, big part of this is just going to be bones and stuff. Incisors, canines, cheek teeth, premolars, molars. Heterotrophic, hind limb. Oh, yeah, okay. Forelimb, hind limb. Because we're getting into the tetrapods or whatever, I don't know. Internal fertilization, intestine, large, small. Jaw, buccal chamber, pharynx. Joints, interesting. Ooh, okay, I'm gonna like the joints. The joints are cool. Suture, symphysis, diathrosis, keratin, bill, claw, hoof, feather, kidney, larva, larvacia, larynx, lateral line, limbs, plantigrade, digitigrade, unguligrade, ungulate probably, liver and gallbladder, locomotion, arboreal. Oh, I don't know this. Arboreal, cursorial, fossorial, natant, saltorial, and volant. Yeah, we'll need to remember that. Lungs, mammalia, mandible. Whoa, I don't know this. Dentary, mandibular condyle, angular condyloid, coronoid. I don't know any of that. Mesenteries. I don't know that. I know it's the middle something, but I don't know what that is. Mesoderm, mesodermal blocks, myotomes, sclerotomes. I don't know what those are. Mesodermal blocks and endodermal pouches, pharyngeal arches, metaphrodium, morphogenesis, morula, motile, mouth, multicellular, muscle, circular and longitudinal, muscle skeletal, smoother cardiac, myotomes, myomeres, nervous tissue, neurulation, notochord, operculum. Do you have a operculum? Yeah, we do. Organ, ossification. A ton of this. This is all just anatomy, man. Somide, siphon, skeleton, skull, spinal cord, spine, spiral valve, spleen, stomach, suspension feeding, swim bladder, teeth and pharyngeal, tetrapod, thoracic cavity. Okay. Alright, that's pretty cool. It's like the main thing is going to be remembering all these bones, I think. Alright, let's get back to this. What is A? What the hell is A? Mm-mm.
This is a neuron, which has four main parts. The dendrites receive information, the cell body processes and integrates that information, the axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions, or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron, in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately minus 70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electrical gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces, such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion, i.e. voltage-gated sodium channels, as well as voltage-gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use. Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from minus 70 millivolts to the threshold voltage of minus 55 millivolts, or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electric.
Okay, it triggers what? The voltage-gated sodium thing?
States: open, closed.
So
Voltage-gated sodium channels have
open, closed, inactive.
three states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive.
Damn, that's actually insane.
As it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels.
The positive?
Membrane potential
Is this also
into the cell. The change in membrane potential also opens the voltage-gated potassium channels, though they
Yeah.
open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period, the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place. And the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate opens. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period, because while the sodium channels could open, it would take a larger than usual stimulus to reach threshold, because the cell is hyperpolarized.
Dude,
due
this
to the
system
potassium
is
still
so
leaving
well tuned. It's actually
cell.
insane.
The amplitude of the action potential for a particular neuron, that is, the maximum voltage in one neuron during an action potential, never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain, and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheaths around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signal appears to jump along the part of the axon covered by the sheath. In the peripheral nervous system, the sheaths are formed from glial cells known as Schwann cells. There are small gaps between Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous system, the sheaths are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a graded potential, and a stimulus above the threshold creates an action potential and the neuron fires.
They didn't really explain anything about the
What if everything you did, thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume. What's 1,000 times thinner than a piece of paper, more numerous in you than grains of...
Bro, chill. Like, my understanding is it kind of... It's a wave, right? Okay, imagine you trigger an action potential at one point in the axon. Voltage gated sodium channels open. Sodium rushes in. Inside becomes positive. Membrane there goes from about minus 70 to plus 30 millivolts. Positive charge spreads sideways, local current. That plus charge on the inside of the segment A flows to the neighboring region B, which is still at minus 70. So inside, okay, that makes sense. I get why it does that. Calcium vesicle fusion line at the presynaptic terminal.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neuron are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is
That's actually pretty crazy.
nanometers wide.
40 nanometers, that's
The neuron
actually
where the
pretty
signal is
nuts.
initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it.
Dude, okay, this is actually so cool. All right, I'm into this stuff, man. This is really amazing. Okay, so let's plot the voltage over time and get a picture of this thing. So first we see a deviation here. Okay. So let's go back to the...
What if everything you did
Here we go.
Contribute to establish when thinking about concentration of rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately minus 70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electrical gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels open in response to physical forces such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump.
Okay, so the graded.
This is accomplished primarily when ion channels open and a graded potential occurs. Neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use. Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from minus 70 millivolts to the threshold voltage of minus 55 millivolts or higher, this triggers an action potential at the axon helic, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative. And more positive as it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell.
It also causes the potassium,
The
right?
change in membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close. For a brief period, the membrane potential is hyperpolarized. It's more negative. Than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place, and the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential, no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards. Absolute refractory period, the period of time when a nerve cannot fire another action stimulus at all. The inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period. The period of time when a nerve cannot fire another action potential, no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate opens. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period.
Okay. All right. I think it makes sense now.
Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic.
At the presynaptic terminal and action potential, there's one key thing. AP arrives at presynaptic terminal, voltage-gated calcium channels open, calcium rushes in, calcium triggers vesicle fusion exocytosis. Yes, so exocytosis itself raises neurotransmitter concentration in the synaptic cleft. How that chemical step becomes an electrical step. On the postsynaptic membrane, you have receptors that are basically ion channels waiting for that specific neurotransmitter. For an excitatory synapse, think glutamate, AMPA receptor, acetylcholine, these ligand-gated ion channels. Neurotransmitter diffuses across the cleft. Neurotransmitter binds postsynaptic receptors. Receptor opens its ion channel. I see. Okay. How long have we going? 2 hours? Damn, it's already been 2 hours? What the heck? Synapse, functional connection between a neuron and another neuron or an effector cell, muscle or gland. The synaptic cleft, a slight gap between the presynaptic axon terminus and the postsynaptic cell. Series of action potentials conducted to axon terminus causes exocytosis of vesicles containing a chemical messenger neurotransmitter into the synaptic cleft. Neurotransmitter binds to a receptor protein on the surface of the effector cell and turns on the receptor. The intracellular portion of the activated receptor causes a response in the postsynaptic cell. Oh, that's it? Oh, we did it. Let's go. Let's go. So now we're finally at the video, I think. So we still need to do this voltage one, right? Oh, okay, here we go. It's probably just going to be the... Wait, what's up with that? That's weird. Wait, minus 10 is the threshold? I thought minus 55 was the threshold. Oh yeah, here we go. This is the standard AP. Mm-hmm. They're called EPSP. Excite Post Synaptot. I think the scale of this is wrong, because if it hits minus 55 no matter what, I think it opens. Yeah. All right, cool. So we're doing sensory systems, but I feel like, am I missing something? Is this the videos? So wait, hold on. So we did this, of course. Let's see what is in this. I think we're on... Oh, but this is 13B. We didn't do this one. This is the wrong one. I was like, what is going on? Okay, so this should end with...
Pituitary. The epithelial part of the pituitary produces no effect.
Right.
Where a deep transmitter, it might cause an excitation of ventilation pathway. There's nothing intuitive about it into it.
Okay, so I didn't really watch all of this, but I feel like I get it. There's probably some
inhibits you
stuff
from
that I
going
didn't exactly do, but I don't really have time to watch that one. But let's watch this one maybe.
So we left off talking about cellular communication and the endocrine system and the neuronal system. Let's talk a little bit more about how neurons actually. So reminding ourselves, first of all, the structure of a neuron, a nerve cell, the cell body, where the nucleus and most of the organelles are, where most of the protein synthesis is happening, the dendrites that greatly increase the surface area to receive signals, and then the axon that conducts those signals. So in dendrites receiving signals, the cell body, where the genetic machinery for the cell and synthesizing all those neurotransmitters, all that cytoskeletal structure, all those gate proteins, that's all regulated there. And the axon, the part that actually conducts the electrochemical impulse. And again, remember, neurons are not wires conducting electricity. These are a series of electrochemical events. And then the terminus, where we're transmitting that message to target cells. So that's the producing of the signal molecule that's going to diffuse or bind to a receptor and stimulate or inhibit another cell. And so he said, in order for an axon to work, it needs to be polarized. There needs to be this concentration gradient and therefore a charge gradient across the membrane. And so primarily the function of the sodium-potassium pump, pumping sodium ions out, potassium ions in, more sodium out than potassium in, creating a charge gradient and multiple concentration gradients. So we can see that the potassium is very high inside the cell relative to outside, so its concentration gradient is wants to leave. Sodium and chloride very high outside the cell relative to inside, so their concentration gradient they want to enter. And whereas the fixed anions, things like proteins and nucleic acids, are fixed inside. They're not going to be able to cross that membrane. And so this voltage then, which we would then call our resting potential, is this difference in charge created by this difference in ion concentrations from one side of the cell to the other. And so if we just look at a voltage scale here, measuring in millivolts over time, at resting potential, the voltage would just continue along at that. I
feel like this is all pretty.
Even down the axon.
Right.
The propagation of the action potential is the same down the axon. So for all of these neurons then, the integration center, the interneurons, the motor neurons stimulating effector, those neurons then, whether they fire or not, is the summation of the generator potential of do you make it to threshold or not, stimulated by inhibitory and excitatory neurotransmitters. But when we talk about the sensory system, now all of a sudden, how do you stimulate a neuron with something other than neurotransmitters from another neuron? These neurons are now unique, that somehow they have to generate that generator potential in response to some change in the physical environment, not a neurotransmitter from another neuron. So the senses then are varieties of different specialized neurons that have somehow been designed to create generator potentials in response to physical stimuli of various types, as opposed to being stimulated by another neuron. So these are the only neurons stimulated by something other than another neuron.
Yeah, so the input neurons, yeah.
So these sensors then,
That's
these sensory receptors, are what we call transducers, and that term is we use that in electronics as well. It's how you take a electrical signal that's produced in response to light or touch or temperature or whatever it is. So we use the same vocabulary here for our sensory system. So a transducer then is a structure that converts a stimulus, some change in the physical environment, to a graded depolarization.
I'll write that down. Transducer is defined as a structure that converts a stimulus into a graded depolarization.
So we're transducing that physical stimulus into a biological signal. So physiological transducers are usually derived from specialized dendrites, then often associated with modified epithelia. So either it's the dendrites themselves or the association of dendrites with special epithelia that create these physiological transducers, these structures that can somehow take the presence of a physical stimulus and create a generator potential. And we have various mechanisms of categorizing these types of transducers. So exteroceptors are external receptors. They're responding to stimuli from outside the body. So things from the environment that our body is being impinged upon, it's perceiving or responding to those things. And so that can be either passive, where you're feeling temperature, touch, light from the environment generated by something else, or active, where the animal produces the signal and then sees how that signal is changed by the environment. So now they're actually creating the signal and then seeing what's the difference in the signal from when I made it to when it comes back to me. And so things like sonar, so echolocation, right? The bat actually makes the sound and then listens for the sound that it made. Or a flashlight fish that produces light and sees what happens to the light. Or electric fields. You have electric fish that create electrical fields and see how those are changed by the environment around them. So those are called active senses. Active senses, the animal produces the stimulus itself. And then there are interoceptors where your body is responding internally. So again, we have all these homeostatic mechanisms in our body. We need to be able to perceive what's going on in our body. So interoceptors sensing the internal environment of the body. So that's one way to characterize transducers. Are you responding to external signals from the environment, external signals from the animal changed by the environment, or internal signals? We also categorize them based on the basic mechanism in how are they taking some physical stimulus and producing a generator potential. And we put them into some, like we always do, we categorize things because there's so many thousands of different types of receptors out there, sensory receptors, that we put them into categories to make it easier to try to get our heads around how they're working. So chemoreceptors, the stimulus is going to be the presence of some chemical signal. Mechanoreceptors, some physical touch or some actually bending the cells. Thermoreceptors, responding to
Okay, so
temperature.
first we have the active, right? Or the extero, intero, then of extero. We have passive. Then mechanisms. We have the chemo, the mechano.
One way to characterize transducers are you responding to external signals from the environment, external signals from the animal changed by the environment, or internal signals. Being a generator, sensory receptor, this is going to respond to other types of electromagnetic energy other than temperature or light. They're able to respond to other types of electromagnetic signals. So you've probably heard, you've got your five senses. You have way more than five senses. So first of all, let's talk about the general mechanism of how these transducers are working. And again, going back to thinking about how do neurons get stimulated? And they get stimulated by opening and closing sodium gates, potassium gates, chloride gates. So these are cell membrane bound proteins that are somehow being stimulated to either open or close. Okay, so all of these mechanisms of transduction are somehow opening or closing gates that are resulting in accumulations of IPSPs and EPSPs, right? Although it may not be synaptic in this case, so you wouldn't call it that. So a chemoreceptor, that's not too hard to get our heads around because we already know that neurons respond to neurotransmitters. Those are chemical signals. So the neurotransmitter binds to a receptor. You bind to the receptor that opens or closes a gate. So in these chemoreceptors, then, it's some other substance other than a neurotransmitter, but as long as you have a receptor that binds to it, and by binding to it, opening or closing a gate, you've got a chemoreceptor. So your chemical stimulus binds to a specific binding protein in the cell surface, open ion gates, depolarization, basic cell communication mechanism. And so we have both internal and external chemoreceptors, right? Your interoceptors, right? How do you maintain your blood sugar, your blood fatty acid, your blood pH, right? Obviously, your body is able to sense when you deviate from the ideal set point. You need have receptors. What sometimes isn't recognized is nociceptors. So something that's noxious is something that bothers you, that irritates you, right? So nociceptors are pain receptors. And so it's surprising to many people that these are actually chemoreceptors because cells that are being damaged release certain molecules. And these nociceptors have receptors to those stress molecules. So you're no pain, no gain, right? No pain, no damage. Damaged cells release chemicals that stimulate nociceptors, which your brain then perceives as pain.
Oh.
And then we have external chemoreceptors as well, right? We smell things, we taste things, we are responding to chemicals from the environment, either by traveling through the media around us or in something that we're touching directly. So chemoreceptors, okay, mechanism that as long as you can build a variety of different receptors, you can respond to a variety of different chemicals. So that's not too hard to understand once we've already understood how basic cell communication works. Next, we have mechanoreceptors. And so we can kind of get our heads around this too, because we're thinking about proteins in the membrane of the cell. If you bend the membrane, you're going to be bending those proteins. And so physical distortion of the cell membrane is causing these ion gates to open. Right? So these are proteins that are designed that when you stretch them in a certain way, they're going to cause ion gates to open. So it's not that you're making the...
Oh, wow. That's amazing, dude. The fact that that's crazy, dude. este pendejo no aceptó el 1.400.000. Y esos eran cortes locales, estatales, todo ahorita van a lo federal.
Stretching the membranes of those specialized cells. So things like touch or things pushing on you, pressure, you can feel that because you're bending the cells in your skin. But then we have a couple of our special senses, our sense of hearing and our sense of equilibrium, that has special mechanoreceptors called hair cells. And they're not hairy, but they just look like, it's sort of like when we talked about the lining of the intestine, the brush border, right? They look brushy. These look like hairy under the electron microscope, but these hairs are actually cilia. That when you bend the cilia, you're distorting the membrane of the cell and therefore stimulating the cell.
Bro,
and so these are some
that's
special
amazing.
sensors that have special protrusions on them that you bend those protrusions, and that stimulates the cell. Okay, so mechanoreceptor, you're physically distorting the cell. That opens gates. You depolarize, you get generated potential. Thermoreceptors. Again, this one shouldn't be too hard for our head to get around either because we know that proteins change shape when you change temperature. Right? That was that whole thermoregulation thing. So if the temperature changes, the proteins are going to change shape. These particular proteins, they change shape in such a way that open ion gates, depolarize, generate potential, nerve impulse.
See, this stuff is actually really interesting to me because it's like all of this stuff seems so ripe for design, optimization, and engineering of different random biological systems. Like, oh man. The thing is, it's funny because it's like, yeah, life figured out, basically you just want homeostasis. So pretty much most of it is going to be like, maybe like local positive feedback, but like global negative feedback or something like that. Like you still need, you can't just be homeostatic all the time. Like you need to respond to stimuli, but, or I don't know what I'm trying to say. I'm just fucking.
And again, we have internal receptors measuring our body temperature, especially those of us that are homeotherms. As part of our homeostatic mechanism, we have to be able to detect when the temperature changes. And then our cutaneous senses also have, right, we feel the environment around us. Is it too hot or not hot enough, which we perceive as cold, right? Cold is not an entity. Cold is not enough heat. So again, thinking of ways that you can bend a protein. You bend a protein, you're going to change its activity. You change its activity in such a way that opens a gate, you can get a nerve impulse. Now we start getting into the electromagnetic receptors, and they're not quite so clear-cut in terms of why would light, for example, cause a protein to change shape? And the answer is, of course, you have to have a pigment. You have to have some sort of molecule that absorbs that light energy in such a way that the protein associated with that pigment is changed in turn. And so these photoreceptors then have these light-absorbing pigments associated with these proteins. When they absorb this light energy, it modifies the protein. It modifies the protein then causes gates to open and close. And interestingly enough, and we'll talk more about this probably next lecture, but in many of the cases with photoreceptors, instead of actually depolarizing and stimulating neurons, they actually hyperpolarize and turn neurons off. And we'll talk about the so-called dark current, where they're firing in the dark and you stop them firing with light. And so again, detecting external signals. So the rods and cones in the retina of our eye then would be photoreceptors. And then we've certainly been able to detect that there are many animals out there that respond to other types of electromagnetic radiation. So electromagnetic fields, infrared radiation. In a lot of cases, we're not really sure what the transduction mechanism is. Somehow, their proteins are associated with structures, usually some metalloprotein, some metal protein complex, that's somehow detecting these fields, so that the presence of these fields are causing these proteins to change shape, open gates, depolarize, and cause neurons to fire.
Dude, you could build a freaking antenna. What if you could listen to the radio with just a bunch of proteins?
So the mechanisms are often a little shakier on these, but definitely measured their effect, that we know that these animals are able to detect these types of stimuli in their environment. Okay, so those are our basic mechanisms of transduction. How do they actually work at the organism level? And so, as we just alluded to, then we need to create a generator potential that makes it the threshold. So you
The thing is, I don't know if it would be even fast enough.
stimulate it, you get a little bit of depolarization, that's not enough. You stimulate it a couple of times, and you get closer, you stimulate it. And finally stimulate it enough times, you make it the threshold, boom, you get an action potential. Once you get an action potential at the hillock, you get a nerve impulse conducted down that axon. Okay, so these receptor potentials or generator potentials are cumulative. Now, we said the action potential is all or none. So how do you tell the difference between a strong signal and a weak signal? Well, there are various mechanisms that happens. There's no difference in the action potential between a strong signal and a weak signal. And so you could have different receptors that respond to different stimuli, so they may have different thresholds. But more commonly, what you do is, it's not the size of the action potential, it's the frequency of the action potential. That if I get a signal and it's still at threshold, I'm going to get another signal. And if it's still at threshold, I'm going to get another signal. So how frequently am I making action potentials? Right? Every time I get one action potential, which we're talking about milliseconds here, an action potential at the hillock, I'm going to get a nerve impulse travel down an axon. If I get another threshold stimulus, a generated potential, then I'm going to get another action potential, series of action potentials down there. So the intensity of the signal is transmitted by the frequency of action potentials, not the amplitude, not the size of the action potential. Okay, so here, for example, we get a stimulus, and as long as the stimulus is there, you get action potentials. Okay, so that's called a tonic response. As long as the stimulus is present, you're going to get nerve impulses firing down there. Right, so this is the frequency of action potentials. Now, a lot of times your body's not so interested in chronic stimuli. It wants to know when there's changes in the environment. And so in that case, it has what's called phasic receptors. And phasic receptors respond more to change. So when the stimulus first starts, you get a lot of action potentials, but gradually it peters off. You stop making action potentials. And so the stimulus is still there this whole time, but the frequency changed. And so that's one way your body can tell changes in the environment, as opposed to things that are just going on all the time. So the stimulus intensity then is transmitted then by the frequency of action potentials. So here you get this receptor, so you get a gentle, so a touch receptor, for example, you touch it a little bit, you get action potentials. You touch it harder, and you get more frequent action potentials. Okay, so this frequency modulation, that's so we're FM, not AM, right? We're not changing the amplitude of the action potential, we're changing the frequency of the action potential. And so when the receiver cells then are telling it's a stronger stimulus, because it's getting action potentials more frequently, not bigger action potentials, more frequent action potentials. So that's the main way that the body communicates intensity of the signal. Now, sometimes you also get what's called receptor recruitment, meaning that the stronger the stimulus is, the more receptors you're going to stimulate. So here, for example, gentle pressure, you're stimulating this part of the skin, you're stretching this part of the skin, so those are the receptors that are firing. You push it harder, and now you're stretching more of the skin, and so your brain's going to be receiving signals from more signals. It's going to assume that's a stronger signal. Okay, so that's another way that the brain can receive information that it will interpret as a stronger signal. So this leads to another central idea of sensory systems. Okay, so this is important. We're going to come back to this concept multiple times. This idea of sensitivity versus acuity. And like with all things in life, there's always trade-offs. So
Okay, here's where I... Okay.
do you want a more sensitive system where I can detect the presence of a very weak signal, or do I want an acute system where I can tell details of that signal? Those are the trade-offs you have typically in how sensory systems work. Okay, so what do we mean by that? Well, we said that, again, to get a generator potential to threshold, we have to add together signals. Now, we can add them together either because we're stimulating the same point multiple times, or we can be stimulating multiple dendrites once. So we can add them up either in time or in space. So adding them up in space, you're going to be more sensitive if you've got more dendrites receiving signals. Okay, that's called convergence. How many sensors do you have converging on that sensory neuron? Right, converging. together. So this, again, another meaning of the word convergence. And so here, for example, we've got a bunch of neurons with low convergence. There's very few receptors converging onto that neuron. Here we've got some neurons with high convergence. There's lots of sensors converging on that neuron. Right? So this is high convergence. This is low convergence. Okay. So now with low convergence, what are the odds of stimulating this receptive field? So the receptive field is that area of the skin or whatever your sensory system is that's stimulating that particular neuron. Okay. So any stimulus that falls in this receptive field is going to stimulate this neuron if it makes the threshold. Whereas if it falls in this receptive field, it's going to stimulate that neuron. And so if you have low convergence, you've got small receptive fields. If you have high convergence, you have big receptive fields. So now you've got a whole big area accumulating stimuli to say, do you make it to threshold to stimulate that neuron? Right? So it makes it easier to detect a signal because it's like having a big satellite dish as opposed to a little rabbit ears antenna. Okay? So you're more sensitive with high convergence. More high convergence, you have a big receptive field adding up stimuli to make it to threshold. Whereas with a low receptive field, you've got to have a strong enough signal hitting you in that tiny little field that you're going to add it up only if the signal is very strong to stimulate that neuron. Okay, so higher convergence, bigger receptive fields, higher sensitivity. So high convergence, high sensitivity. Okay. Now the problem is with a big receptive field, though, is that all you know is that signal, that stimulus came somewhere in that receptive field. You don't know anywhere. You don't know exactly where.
So it's like
whereas here, because the receptive fields are smaller, if I stimulate that neuron, I know it came from that receptive field. I can more pinpoint more precisely where that stimulus came from. That's called acuity. Being able to tell
convergence is kind of like gain.
So, for example, if these are all stimuli, they're falling on your skin.
So there's a trade-off between.
And I've got let's say it takes. You have to hit it twice to say that this is a signal. You're going to accumulate enough signals to fire that neuron, to stimulate that neuron. Where if this is how fast they're hitting, then with one wave, then this is going to stimulate this enough to fire that neuron. It's more sensitive. Where these, one wave, you're not getting two hits in any receptive field, none of these neurons are going to fire. But if I do stimulate this, if one hit is enough to stimulate it, then here I can
just
tell that
spatially you have a trade-off. You can either be
three hits.
high
Here,
convergence,
three different receptive fields, stimulating three different neurons.
Low
We're here, three stimuli hitting one
convergence,
receptive
high
field, so stimulating one
acuity.
neuron.
Yeah, makes sense.
I'm getting one signal to the brain as opposed to three different signals to the brain. So the brain just knows that I got a signal from somewhere in the receptive field. Where here I'm saying I got these three that got hit.
But
so I know
like for this, you need to have like a strong signal to activate it.
Here, I don't know where it's hitting, somewhere in here.
Yeah, makes sense.
So I can tell detail because I have more parts of the brain being stimulated by these receptive fields. Right? These are all stimulating cells in your brain that are then able to tell that, oh, I got a hit, oh, I got a hit too, I got a hit too. So I must have gotten three different stimuli. But here it's like, I got a hit. I only know I got one stimulus. Okay, so that's reflected then into how many cells in the brain. So here in the brain, that this neuron stimulating this cell in the brain, this cell is stimulating this. So you're stimulating all these different cells in the brain. But here, that's stimulating one cell, that's stimulating another cell. Not a whole lot of brain involved with processing that information. And so you can actually map the sensory cortex. In the cerebral cortex is the outer part of the brain, and the cortex the outer part.
Whoa, what the fuck?
The cerebrum is where you perceive these senses, and so this outer range, the cortex of the brain, is proportional to the acuity of the system that's responding to. So you can see here in your hands, you have high acuity, so proportionally the part of your brain that's receiving signals from the brain is much bigger than from your shoulders. Okay, that's why something is crawling on your shoulders, you can feel it's there real well, but you can't tell what it is. It's like, where on your hands, it takes a stronger signal to stimulate it, but you can tell really fine detail with your hands. You have high acuity senses in your hands, high sensitivity in the back of your neck. Right? That's why you're more ticklish in those places, because it feels something there, but I don't know what it is. With my fingers, I can tell exactly what that is. Okay, so this trade-off then of convergence versus sensitivity and acuity. High convergence, high sensitivity, low acuity. Low convergence, low sensitivity, but high acuity. Okay, that's going to be a repeated theme in all the different senses that we're talking about. Do you need to detect the presence of a weak signal, or do you need to be able to tell detail in the quality of that signal? Which then comes to the point that it's the brain, it's the cells in the brain that are actually perceiving what it is, right? It's all nerve impulses. There's no difference in a nerve impulse from a touch receptor and a thermoreceptor or a pain receptor. Right? It's all just a series of action potentials. So how can you tell the difference between something touching you and something burning you and something that's too cold? And it's because what cells in the brain are being stimulated. And so this is called the law of specific nerve energies. That is actually the cell in the brain that's determining what you perceive it to be. So when they say it's all in your head, it is all in your head. So anything that stimulates that cell in your brain is going to be assumed by your brain as being coming from that part of your body, and the type of stimulus that would stimulate that part of the body. So if this is a cell that normally receives a pain signal from that part of your hand, then anything that stimulates that cell is going to be perceived as if something's hurting your hand right there. So any stimulation of a sensory neuron is perceived according to that nerve's adequate stimulus. And adequate stimulus is the normal type and intensity of stimulus that would stimulate that neuron. So if a brain cell is stimulated, it's assuming it's because of the normal thing that cell responds to. Okay, so sometimes if you stimulate cells in your brain, so if you actually stick electrodes in your brain, it can feel like things are touching your body when they're not. And the type of stimulus. So you get punched in the eye, and all of a sudden you see stars, right? Well, where did that light come from? There was no light. You stimulated cells in your eyes that normally respond to light, therefore the brain assumed that there was light. Or you get congestion, and your ears start ringing, right? Where's that sound coming from? There is no sound. But congestion is impinging on nerves that normally respond to sound, therefore your brain assumes that there's sound. And so you hear sound that's not there. Or phantom pain, amputee, somebody gets their hand cut off, and now it feels like their hand hurts. How can their hand hurt? It's no longer attached to their body. Well, the nerves that used to come from the hand are still there, and they're awful irritated because they just got cut. So it feels like your hand is there. So phantom pain, this pain in an organ that's not even there anymore. Right? But the cells in your brain are still there.
Wow.
Or sometimes, right, somebody's having a heart attack and they feel this pain radiating down their left arm. Right? What's the matter with their left arm? Nothing. But the nerves that normally come from the left arm are impinging on the nerves coming from the heart. And so it's feeling like the pain is in your arm, but it's not your arm. The pain has been referred from where it actually is coming from to feeling like it's coming from somewhere else. Or even spices, right? You have hot, spicy food, right? How did the spice make the food hot? It's stimulating thermoreceptors in your tongue, so your brain perceives that as if it's hot. Okay, so it's what cells in the brain are being stimulated are then assumed and interpreted by your perceptions as the thing that normally would stimulate that sensory cell in your brain.
That's so cool, man. That's really interesting.
Okay, so we talked about different categorizing sensors based on what type of signal they respond to, that they're transducing. We also can categorize them based on the path they take from wherever that sensor is to the brain. And so with our somatosthetic senses, so soma, remember, means body. Aesthetic means feeling, right? Something that's aesthetically feeling is something that makes you feel good. Something that's an anesthetic is without feeling, takes away feeling. So aesthetic means feeling, soma, body. So these body feeling senses.
Aesthetic.
And these include your cutaneous senses in your skin, the visceral senses, the stretch receptors in your gut, things like that. The proprioceptors, the positioning receptors in your muscles and joints. Those are all what we call body feeling senses. And they travel from your sensors to the brain via what we call by your spinal nerves. So the sensory neurons from these receptors go via the spinal cord to the brain. Whereas the special senses travel via cranial nerves, so they're plugging directly into the brain. So things like your taste, smell, hearing, equilibrium, vision, right? Don't you know the five senses? These are all traveling via cranial nerves, and so these tend to be in your head, right? That's why we're talking about cephalization, all the sensory structures in your head. And so they're traveling directly into the head. Okay. Now the somatosthetic senses are interesting because of the way they actually get into the spinal cord. So from the spinal cord, right, that hollow dorsal nerve tube that we just talked about in lab, is both sending out motor impulses to your muscles and receiving sensory information from your sensors. The sensory neuron are traveling in often a common conduit along with motor neurons called mixed nerve. So you might have sensory neurons and motor neurons traveling in the same cable. They're not talking to each other, they're just traveling in the same cable. But before they reach the spinal cord, that's when they diverge from each other. So the motor neurons come out the ventral roots of the spinal cord. The sensory inputs go in the dorsal roots. So we call these little extensions of the peripheral nervous system off the central nervous system, the spinal roots. Okay, so motor outputs come out the ventral root, sensory inputs come in the dorsal root. Now these spinal sensory nerves have an interesting shape to them. They're what we call pseudo-unipolar, which means that the dendrites don't converge onto the cell body, they converge onto the hillock. So here's all those dendrites. Those converge right onto the hillock, and then the axon just passes by the cell body off to the side.
Oh, whoa.
So the cell bodies are located in this ganglion right here outside the spinal cord. So it's called the dorsal root ganglion. It's a collection of cell bodies right here. So the dendrites are way out here, the nerve impulse travels down the axon, just bypasses right by the cell body into the spinal cord. Okay, so
why do you even have the cell body? Why not make it like a red blood cell and have it be like a...
Now, there's a little bit... spinal nerve. Nerves are bundles of axons, then your sensory inputs, your motor outputs. Now, this is a little busy, but there's a central pattern here you should recognize. That for these somatosthetic senses, these spinal nerve transmitted impulses, we said that the part of the brain that's receiving the signals is called the sensory cortex, the cerebral cortex region, a particular region of the cerebral cortex, the sensory cortex. So that's the outer part of the brain here. Now, between the sensor out in your peripheral body somewhere, and that sensory cortex, there's actually 3 neurons. So there's 3 neurons in the pathway, so it's not a direct pathway. So the initial neuron, the one coming from the sensor, comes into that dorsal root into the spinal cord. Okay? There it's going to synapse, so it may come in this case here, it's coming in and descending up the spinal cord and stopping here. Whereas in this case here, it's coming in and stopping right away. But it's going to synapse with an interneuron. And that interneuron actually crosses over to the other side.
What is the left and right side? What is going on here? This diagram is so confusing.
And travels up to the brain on the other descent. There's 3 brain here. Senses, these spinal nerve transmitted impulses. We said that the part of the brain that's receiving the signals is called the sensory cortex, the cerebral cortex region, a particular region of the cerebral cortex, the sensory cortex. So that's the outer part of the brain here. Now between the sensor out in your peripheral body somewhere and that sensory cortex, there's actually 3 neurons. So there's 3 neurons in the pathway, so it's not a direct pathway. So the initial neuron, the one coming from the sensor, comes into that dorsal root into the spinal cord. Okay? There it's going to synapse, so it may come in this case here, it's coming in and descending up the spinal cord and stopping here. Whereas in this case here, it's coming in and stopping right away. But it's going to synapse with an interneuron. And that interneuron actually crosses over to the other side and travels up to the brain on the other side. It's called contralateral, opposite side. Okay, so the original spinal nerve comes into the spinal cord and stays on that same side of the spinal cord. The second neuron crosses over to the other side and then goes up to the bottom of the brain called the thalamus. That's where it then synapses with the third order sensory neuron that actually goes to the sensory cortex. Okay, so there's lots of variations here, but the first neuron, the first order sensory neuron goes from the sensor to the same side of the spinal cord. The second order neuron crosses over to the opposite side and goes up to the thalamus. The third one goes from the thalamus to the sensory cortex. So the part of your brain that's responding to the stimulus is on the opposite side of your body from where the stimulus came in. And this is all part of the filtering mechanism that your body uses to, number one, make sure that it's not just random electrochemical noise it's listening to, it's actual real nerve impulse. And then the thalamus also acts as a filter that chronic information is often filtered out and not bothered to be transmitted. It's like right now there's a heater blowing in the other room, but did I hear the noise the whole time? No, because it's blowing all the time. I just kind of tuned it out. The thalamus is part of your mechanism that you use to tune it out, right? So stuff that's just kind of chronically going on all the time, you tune it out, right? It's stimulating your sensors, but your brain doesn't need to know. The thalamus makes that decision. It's like, nah, we don't need to know that. Okay? So all these different sensors are coming in different levels of the spinal cord. But the first order is staying on the same side. The second one crosses over to the opposite side and goes to the thalamus. The third from the thalamus, then that's the filter. Third from the thalamus to the sensory
That's
cortex.
pretty cool,
On
actually.
the opposite side of your body.
Thalamus
where the
is
sensory
crazy,
signal
man.
actually came in. Okay. So those are your somatosthetic senses. So again, whether it's a proprioceptor, a cutaneous receptor, a visceral receptor. These are the paths those are traveling on. So looking at your cutaneous senses, so your skin, again, the largest organ in your body, and it's full of sensory structures. And so we can see different.
I'd like to draw that diagram. That's pretty amazing. Mediated by dendritic nerve endings of different sensory neurons, mostly in the dermis. Some in the basal stratum of epidermis. Thermoreceptors, nociceptors, mechanoreceptors, free, encapsulated touch and pressure.
Levels of this. Most of these are mediated down here in the dermis and hypodermis. Right?
The hair
These are
one
dermal
to
parts
me
of your
is
skin.
pretty
Those are
cool.
epidermis, which is
I really
the
like
derived
that.
parts.
Yeah, some
And so these are typically
mechanoreceptors
the
in the hair
dendrites of these sensory
movement.
neurons
That's so cool.
that are specialized to respond to temperature, chemical, or
Because the
physical.
cool thing about a long hair is that given the stiffness of it, I don't know how, but whatever, but you have a lever. So you can actually amplify that. You can be really sensitive to small changes at the, end of the hair.
Stimuli. So again, we got thermoreceptors that respond to heat and cold, nociceptors that respond to distress molecules created by damaged cells, your pain receptors, and then mechanoreceptors that are responding to touch. And we have a variety of these because your skin does perceive a variety of types of touch and pressure. So there are free nerve endings that are kind of more exposed, and these often penetrate all the way up into the epidermis. And then there are, and some of them are slow adapting, the tonic ones, so that as long as the stimulus is there, they respond. Others are phasic. And then there's some that are wrapped in this epithelial tissue, these little capsules. And these tend to be more the phasic type that respond to changes in touch as opposed to things, right? You don't want to be, your clothes are touching your skin all the time. You don't want to be necessarily responding to that. It's like, okay, I felt it when I first got dressed. I don't need to keep feeling it. And then some of them will wrap around like the base of your hair follicles. And so now one of the things your hair does is extends your sensory field out beyond your skin, right? So if something bends your hair, it's going to bend these dendrites and therefore cause a signal. So you extended our sensory field by having hairs in the base of the hair wrapped with these sensory dendrites. So those are cutaneous sensors. Now, how does a bug do that? A bug's covered with an exoskeleton. Well, they've got to have articulated bristles in their skin. And so these are called sensillae, these sensory bristles. And so they're articulated here with the nerve endings then penetrating through that exoskeleton. So that something bends that bristle, it's going to stimulate the dendrites of that sensory neuron. So it's analogous to those dendrites wrapped around the base of your hair follicles. We've got these bristles whose function is sensory.
Oh, because they have an exoskeleton, right? Because that's chitinous or whatever. So they're not going to be able to. They have no dermis or, it's not skin. They can't feel it. I guess.
And so the arthropods have a whole variety of these different types of these things. So you look at an antenna particularly, but anywhere in the body, they can have these sensillae, these sensory bristles, that are different sizes and shapes and responding to all kinds of different signals in terms of a slight touch, a heavy touch, all sorts of things. So we call them hairs and pegs, depending if they're skinny or long. But a whole variety of these things, as well as these kind of perforated discs that can respond to chemicals, as well as pit organs, pore plates. So a variety of sensory structures that are built into this exoskeleton that they're shedding all the time and need to be replacing these components and how they're connected. So here you got a bunch of sensory neurons that are connected then to these structures in the exoskeleton. So pretty amazing that these insects are covered with this armor and can still sense their environment.
Bugs.
So the somatostatic senses include the cutaneous senses, and then those proprioceptors, again, these stretch receptors in the muscles, in the tendons, and in the ligaments that tell the body its position. So within muscles, there's these structures called spindles that tell you if your muscles stretched. Then that keeps the slack out of your muscles, so your muscles aren't just kind of flopping off your body, but they're always maintaining a tension, so that when you need them, they don't have to take up the slack first. They're always have some tension on them, so that they're always tight, so you're ready to use them. And then these Golgi organs and the tendons that prevent you from over pulling, because potentially you could contract that muscle strong enough to pull it right off the bone. The Golgi organs help inhibit the contraction of the muscle to prevent you from damaging yourself by contracting your muscles too hard.
Oh.
And then the ligaments having these stretch receptors that tell you the angle of the joint. So again, part of the information.
Dude, I wonder if people have like really mapped that. That's crazy. Like, you probably can't just take an fMRI, but like, where does that signal end up? That's amazing. Because like, could you recreate just by, instead of having like, just by using a brain scan of some kind, can you really recreate an accurate joint angle of every, whatever, of the whole musculoskeletal system? Like, that is pretty cool.
Or it is, by being able to tell how hard they're stretched, therefore what is the angle.
That's
on that
awesome.
joint. So these are collectively called proprioceptors.
I really like that one.
And then these stretch receptors, again, either in muscles, skeletal muscles, or in visceral muscles, again, looking at these stretch receptors, in this case, in a crayfish abdominal muscle. And so this idea that, again, you get a small generator potential.
Yes, C. elegans does have proprioception, and it has neurons that act as proprioceptors, but they don't look like vertebrate muscle spindle organs. Position feedback. Because I feel like I've seen videos where they do, what is it called? Neuron trajectory model? I don't know, but they like plot in phase space. Because I think they have like exactly a couple hundred neurons or something crazy. But it's like always the same. Like, to me, that's one of the most amazing things about C. elegans. Like, it's always got the same number of cells, basically. Like, how the hell is that? How does that happen?
You get less frequent action potentials, you get a strong generator potential, you get more frequent. Right? The action potentials don't change. It's the frequency of them that changes. So conducting that information by frequency of action potentials, not size of the action potential. Okay, so those were our somatosthetic senses. Now we start looking at some of our special senses, and we can, for example, look at chemoreceptors, which, as we talked about, means you have special dendrites associated with receptors that bind to specific molecules. And so obviously being able to tell, taste, and smell food is of primary importance to animals because we are heterotrophs. We need to be able to feed on things that will give us nutrition. So palatability, does it taste good or does it taste like something that you should be actually eating? Actually detecting the presence of food if you're hunting and looking for the food. And then besides feeding, this is often the way many animals find mates or find their mothers or find a lot of other social interactions is by chemical cues. Those pheromones that we talked about. And interestingly, again, convergence, that things as diverse as elephants and insects use the same molecule as a pheromone to detect the presence of mates. How did that convergence work? And then sometimes not just to recognize, but particularly in social animals, to recognize that if you encounter another member of your species as a part of your tribe, of your hive, do you accept this individual or do you fight with it? So are you going to allow this individual into your nest? Or ants that follow each other on these trails, right? They're using these chemical cues. Obviously, if the chemical is going to be a cue, you need to have a receptor that responds to that chemical. Now looking at some of our chemoreceptors, of course, we have our gustatory senses, gustatory taste, primarily in our taste buds in our tongue. And so in our taste buds in our tongue, the buds themselves are actually epithelial cells. So you see these epithelial cells with these microvilli at the surface, you get the lots of surface area with lots of receptors. And then those epithelial cells associated with those dendrites respond to the same type of chemical signal. Okay, now again, these are special senses, so they're traveling directly via cranial nerves. So the sensory neuron from the taste bud goes to the medulla down in the brainstem, then back to the thalamus again, so the thalamus is also filtering this.
Wait, what did he say?
The surface, you get the lots of surface area with lots of receptors. And then those epithelial cells associated with those dendrites respond to the same type of chemical signal. Okay, now again, these are special senses, so they're traveling directly via cranial nerves. So the sensory neuron from the taste bud goes to the medulla down in the brainstem, then back to the thalamus again, so the thalamus is also filtering this, and then to the different region of the sensory cortex.
Bro, the thalamus seems cool. Like, what is the design there, bro? The thalamus, or chamber, is a large mass of gray matter on the lateral wall of the third ventricle, forming the dorsal part of the diencephalon. Yeah, okay, cool. Yeah, I knew all that. Duh. The nerve fibers project out of the thalamus to the cerebral cortex in all directions, known as the thalamocortical radiations, allowing hub-like exchanges of information. It has several functions such as the relaying of sensory and motor signals to the cerebral cortex, and the regulation of consciousness, sleep, and alertness. Is there a specific function thing? The thalamus has multiple functions and is generally believed to act as a relay station or hub, relaying information between different subcortical areas in the cerebral cortex. In particular, every sensory system, with the exception of the olfactory system, includes a thalamic nucleus that receives sensory signals and sends them to the associated primary cortical area. Whoa. Whoa. That's neat. So, like, often everything goes to the thalamus.
And so you may have in elementary school or middle school had these experiments where you map out where your different types of taste receptors are. Turns out that this is a myth. That, again, you have this preconceived idea. The book tells you this is where you're supposed to taste sweet, therefore you taste sweet there. So you convince yourself that what you're looking for. But it turns out that they're not mapped out like this, that they're not evenly distributed, but they're not in these distinct regions either. Little patches of sweet, sour, salty, bitter, and meaty flavors, the fifth type of receptors. So it's another example of a textbook myth. I told a nice story, so people started believing it. And again, if looking at the arthropods, they have extremely good chemoreceptors, but that means they somehow need to get those chemical signals from the environment across that exoskeleton. So some of their syncytiae are hollow, and they act like little micropipettes that then can sense the chemical composition of things that they're tasting. So, and these don't necessarily have to be on their tongue. Right, these can be anywhere in their body. So a fly, for example, can be walking on things and saying, oh, is that something I should eat or not?
That's actually dope. Imagine you could, like, taste the ground with your feet. I mean, I already do, but, like, imagine if you could. I know what it's like, though.
In the syncytiae of its legs, as well as its mouth parts and its antennae. And so just like we have our 5 taste receptors in our tongue, so the fly has 4 different types of taste receptors in the syncytiae. And so different substances stimulate different receptors differently.
Wow, dude. Imagine the guy that's got to hook up the electrodes to the fly brain. That's wild.
So you have a salt receptor that responds to salt. If you give it something like meat that's got both salt and other things in it, or different neurons will respond. Sugar will stimulate this, honey will stimulate those two. And so again, the brain puts together this combination of how different receptors are stimulated by a particular substance and then come up with a sensation of what does it taste like, right? Just like your brain synthesizes what things taste like from how it stimulates the 5 different types of taste receptors in your tongue. Now, whereas we only have 5 different types of taste receptors, maybe 6, in our tongue, our nose, on the other hand, has thousands of different types of taste receptors. So they can respond to over 10,000 different odors by combinations of these different chemoreceptors that are.
Now let's read this because I really like smell. I feel like smell is really cool. We put olfaction. Bipolar sensory neuron extends. Wait, I never got what is a bipolar neuron? A two-pole neuron. Okay. 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 Ganglia. See, how far are we into this thing? I think we're at this one, right? Okay, we're at olfaction. 5 out of 20? Dude, you cannot be serious, bro. Holy cow, man. We got a lot more to go. I mean, I guess it makes sense that we're going to spend a long time on this, but I was not expecting 5 out of 20. Dude, we got a while, bro. Holy cow.
Is affecting these particular types of neurons. And so it's incredible.
Oh, wow. Actually, that thing is based on the speed. That's funny. I never noticed that. I was always like...
diverse varieties of things you can smell. And interestingly enough, the pathway is much different. The olfactory nerve, cranial nerve number 1, goes directly to the temporal lobes and frontal lobes. It doesn't go through the thalamus. So it doesn't go through that filter. That's why smells will so often get your attention, right? All of a sudden you get that little whiff of the coffee from the other room or whatever it is, and it's like, whoa. Really able to respond to these chemical signals. And that's why, of course, there's a gazillion dollars invested in the perfume industry.
That's actually crazy, dude. The perfume industry. Do you think they're like trying to like... That's crazy. The perfume industry. I never thought about that, though. They're really like... They have the sole advantage of being able to circumvent your thalamus.
Now, in terms of hunting by scent of smell, that requires you can tell where the smell is coming from. And so some animals are really good at that. For example, in the hammerhead shark, one of the advantages of having that weird hammer head is that their nostrils are spread far apart. Now, a shark doesn't breathe through its nose, but it does smell through its nose. And so by having them spread out, it can tell the direction the smell is coming from. Because if the smell is coming from over here on the right, it's going to hit the left nostril before it hits the right nostril. Right, so I'm looking at the bottom here. And so it can tell the direction of smell, so it can kind of swim back and forth and then kind of zoom in on that smell, that scent of prey, that blood trail or whatever it is coming from that area. Or again, insects are amazing at this sometimes. You get this little tiny bug that can track down a female miles away by the pheromones she produces. So these moths in particular, the males typically you see have these huge antennae on them, and that's so that they can zoom in on females from miles away and detect where they are. So two of the most sensitive and specific chemoreceptors known are in the male silkworm moth. Okay, so our first special senses we considered then, transmitted by cranial nerves, were gustatory and olfactory, taste and smell. Those are chemoreceptors. Now we come to the next special sense that we'll talk about. And this is in a part of our head called the inner ear. There are some of these mechanoreceptors called hair cells. Okay, so we alluded to these a little bit earlier. So they're called hair cells because under the electron microscope, they look like they have hairs on the surface. They have nothing to do with hairs. Okay, it's not really hair. Okay, but in the electron microscope, there's these little protrusions sticking out that looks kind of like hair. Okay, but these are mechanoreceptors. These are special cilia that don't move. So they're not beating cilia, they're rigid cilia. And so when you bend those cilia, it causes gates to open and depolarize. Well, actually, it's even better than that because it depends on which way you bend them. If you bend them one way, they depolarize and stimulate the cell. If you bend them the other way, they hyperpolarize and inhibit the cell. So not only detect motion, they detect the direction of the motion. Okay, so these rigid stereocilia, they look hairy, which is why they give that name. So here we can see them a little closer in the position in the inner ear, where there's one large one called the kinocilium in that. And multiples of these smaller, finer ones called the stereocilia. And so the direction that they move, so there's this background firing rate. So under resting conditions, when they're not being stimulated, the neurons just every once in a while depolarize and fire. So that's the resting background firing rate. If you bend the stereocilia towards the kinocilia, you start creating generator potentials more often, and therefore you get a higher frequency of action potentials. If you bend in the other direction, you get less frequent action potentials. And so the brain can tell that these cells are being stimulated because the firing rate's changing. So it's not that they're firing or they're not firing. They're always firing, but it's the rate of firing. If the rate changes in one way, you know the direction of the stimulus came from that direction, that's bending them that way. If the rate of change decreases, you know they're bending the other direction, therefore the stimulus came from that direction. So these are directional mechanoreceptors. Okay, so where do we use them? We say that they're in our head, and they're actually in the inner ear of our head. So one way we use these to detect vibrations in the air, or if you're living underwater, vibrations in the water. And we call these pressure waves and sound waves. So pressure waves move through the media, right? So that's why there's no sound in outer space, because there's nothing to move. But the water or the air moves with these pressure waves. This pressure travels as waves. And so you can use this information. Something caused those vibrations. So you can detect where those vibrations come from and analyze the quality of those vibrations. What does that information tell you? And if you can make vibrations and you can respond to vibrations, you can therefore communicate with vibrations. So how do you create vibrations? Well, with mammals, we have vocal cords. So as we're moving air through the larynx, the upper part of our airway there, we have muscles that can constrict how wide that opening is, and therefore the air causes it to vibrate and make sound. And as those vibrations travel through our head, we then change the shape of our head with our mouth and our tongue and create vocal sounds. So that's why we call our larynx our voice box, because we're creating our vocal sounds. Birds have an analogous structure. It's not quite the same, but it's working the same way, called the syrinx. And so that's their song box instead of their voice box. And unlike us, where we're only making sounds when air comes out, birds, they can make sounds as air goes either in or out. Remember, they have those amazing lungs. Other ways to make vibrations is more, instead of a wind instrument, more of a string instrument, and to create rubbing sounds, so stridulations. So arthropods, for example, have these like rigid teeth structures that they rub together. These might be on their legs or on their wings, and by rubbing them together, can create sounds in those ways. And sometimes it sounds like a deep sound, sometimes a high-pitched sound. I remember when I was in the South Pacific, some of the crickets there make this sound so loud it hurts your ears. Or another way is in, for example, fish, which we'll talk about in lab coming up. Many of them have a swim bladder, which is an air-filled structure that they use for buoyancy. But you have an air-filled structure, you vibrate it, now you've got a sound device by vibrating an air drum, like a drum. And so their sonic muscles, some of the fastest contracting muscles known in vertebrates, where they can vibrate this thing and create sounds. So you have fish called croakers and grunts by vibrating an air drum, like a drum. And so their sonic muscles, some of the fastest contracting muscles known in vertebrates, where they can vibrate this thing and create sounds. So you have fish called croakers and grunts that are making these loud sounds underwater. And water travels through a dense media, or sound travels through water much farther than it does through air. So in characterizing sounds, so these vibrations are traveling in waves, and differences in different sounds is how frequent is the sound. So here you have a high frequency, you're making more waves per second, where here you have a lower frequency, you're making fewer waves per second. So that's the frequency of the sound, which is measured in hertz, which means per second. So how many oscillations per second are you getting? And that's typically the pitch of the sound, is related to the frequency. Whereas the intensity of the sound is the amplitude. How big is that sound? So we talk about waves, we talk about the frequency of the waves, the length of the wave from trough to trough or from peak to peak, and the amplitude of the wave, the height of the wave. Those are the three qualities then of any wave, sound or otherwise. So we talked about ways of making vibrations. How do we then perceive vibrations? And so looking at the mammalian ear, first of all, there are three parts to it: the outer ear, the middle ear, and the inner ear. So the outer ear is pretty much a funnel. And so that goes from the outer pinnae, the actual flap behind the ear, and this canal that then goes to a membrane called the eardrum, the tympanic membrane, tympanic drum. And so this is funneling the sound from a big area into a smaller area, right? So you're focusing the sound. Now in the middle ear, there's a series of three bones that act as levers and fulcrums. And levers and fulcrums help you amplify things. So these are an amplifier. And so the bones are connected up to the eardrum on one side and to another membrane called the oval window on the other side. So you get a little vibration in the eardrum. You get a bigger vibration in the
Amazing.
oval window, right?
Amazing.
The same frequency.
I mean, come on,
but
guys.
it's now
What the heck?
bigger in
That
size.
is just
So
so
it's an amplifier
freaking
device.
incredible.
That's your middle ear. The inner ear is where those hair cells are. This is where you're actually transducing those vibrations to the nerve impulses. Okay, so this is all conduction of the vibrations. You're conducting the vibrations from the air through the inner ear, amplifying them in the middle ear, but they're still just vibrations. Transduction of nerve impulses happens in the inner ear. So the inner ear looks something like this. The part having to do with hearing is this thing here that looks like a snail, and so we call it cochlea, which is, I think, Latin for snail. Now, if we uncoil it, you can't really uncoil it, but just for diagrammatic purposes, if we straighten that out just to see what's going on in there, we see that the structure of this cochlea is a fluid-filled pocket, if you will, with a membrane that separates an upper region from a lower region. So this is fluid-filled, and fluid, again, transmits vibrations. That ear bones connect to that oval window here. So that pushes, every time there's a vibration, it's pushing that fluid in the membrane, therefore creating a wave that travels down this fluid. It travels up and then back. And since liquids can't be compressed, we need to relieve that pressure. So when the inner oval window bends inward, on the other end we have the round window that bends outward. So that's kind of a pressure release system. So you push in here, it's pushing out there. But the idea is that this vibrations then are traveling in waves up the fluid here and back the fluid there. When it does that, it squeezes this membrane in the middle.
Oops. Bro. Fuck's sake.
Now, depending on how oval, when it does that, it squeezes this membrane in the middle. That's where the hair cells are. The hair cells are lining this membrane between these two fluid-filled chambers. So vibrations by the stapes, that's the third bone, in the oval window produces pressure waves that displace the perilymph fluid, that's the liquid in here, within the cochlea. Now, depending on how big that wave is going to tell you how strong the signal is. Where the wave is, how far apart those waves are, is going to tell you the wavelength, which is related to the frequency. So your brain takes all this information. So all these hair cells are communicating, sending nerve impulses to the brain. So the brain knows that vibrations are coming in at a certain frequency and a certain amplitude. And the brain is putting that together, and you're perceiving that as sound. I mean, it's absolutely amazing. The computing power is taking these vibrations and translating them into sound.
Man,
you can tell distinctions of voice, of message, of information, of music. All this information is being processed by the brain.
It's so fast,
which
too.
hair cells are you
Like, I don't get how it's so, like, if you think about,
brain.
like, 10 kilohertz, I mean, that's pretty high, but, like, even, like, 1 kilohertz sound, 1,000 times a second, and it can be transduced totally fine.
Okay, so this is called the organ of Corti. The organ of Corti, then, is this membrane with hair cells that's being squeezed by these waves. Okay, absolutely amazing instrument. So again, your brain is taking this. So again, if you have a sound like this, it's going to be squeezing hair cells here. It's going to be squeezing hair cells here. If you have a sound like this, it's going to be squeezing cells here and here. Right. So the different wavelength sounds are stimulating hair cells at different positions along the organ of Corti. And so the brain can
Wait, actually, this is the best picture I've ever seen in my life. You can just make it longer. Oh, actually, no, because I guess there's a fundamental limit in the density of the mechanoreceptors. So there is like a pretty hard limit, I guess, on the engineering of how high a frequency you can hear. Because after you have the transduction, I mean, it's all just neurons, which is fine. But yeah, so like, look at that. Dude, this is the fucking coolest picture I've ever seen, bro. That is insane, man. Look at that separation. It just separates out all the frequencies. So if you have like a node, oh my God, that's crazy.
detect these and separate them. So it says, because you can be simultaneously getting sounds of different vibrations of different frequencies, the brain can tell that, oh, it's being squeezed with this wave and that wave and that wave, and so I'm going to put that together and perceive it as a sound. It's amazing stuff. So the location of which hair cells are stimulated tells you the pitch of the sound. The degree, right, how wide of an area is it being pushed here, tells you the amplitude of the vibration, how loud is the sound. Okay, so by simply having this information about which hair cells are having their little hairs distorted, your brain can process that information. Into
Bro,
all
the ear is crazy.
Perceive and discriminate about. Amazing stuff. Now, along with the actual ear that's transducing vibrations into sound, there are other aspects of hearing. But again, especially if we're going to use it to hunt with, we need to tell where the sound is coming from. And so that's one of the things that these pinnae do, these ear flaps, is you can tell the difference between sound coming from in front of you and sound coming from behind you, because in order to get into that ear canal, it's either going directly into the ear canal or it's coming through those flaps. Okay, so that's some directionality. Now, some animals are extremely good at hunting by sound. For example, this barn owl here. And so they need to tell in 3 dimensions, where is that sound coming from? And so in terms of from your midlines coming from the right to the left, you use what's called interaural time differences. So the difference in time between the 2 ears. When did the sound hit one ear as opposed to when did it hit the other ear? And they can clock that in their brain to tell that if it hit this ear before it hit that ear, it's coming from that direction, and exactly what angle, the deviation from the midline, is that coming from. And the bigger your head is, that also creates a head shadowing effect. That if it's coming from over here, it's going to be hitting this from the right side, it's going to be hitting my right ear more directly, where my left side is kind of vibrations are traveling through my skull first. And so it can tell that as well. So right-left direction by having this difference in time from when sound hits one ear from the other, and the fact that vibration is traveling directly into one ear canal, but going through the skull to the other one. This again, because sound travels faster underwater. That's why for us, who are used to listening to sounds in air, when we're underwater, we can hear sounds from far away, but we can't tell what direction they're coming from, because it's traveling so fast through the water that it's hitting both our ears at essentially the same time, no matter which direction it's coming from. We can't tell the difference in the time, whereas in the air, we can tell the difference. So we know that the sound is coming from over there or over there. Now, is it coming from a certain angle? That's going to give you the same time difference, whether it's an angle in front of your head or behind your head. And so having these pinnae helps tell, are the sound coming from that angle in front of your head, in front of your midline of your head here, or behind the midline. Then elevation up and down tends to be a little harder, but that's why oftentimes your dog will cock its head. So you talk to your dog goes, whoop, right? So now all of a sudden one ear is above the other. So that interaural time difference can also tell you up and down angles as well as left and right angles. Or with this barn owl, which is amazingly hunting in the middle of the night and finding a mouse buried in the grass, it actually has asymmetrical ears. So you look at one side of its head, the ear is down here, the other side is up here. And so automatically it has this intraoral time difference where it can tell in 3 dimensions
Wait,
what direction the
really?
sound is coming from.
That's weird. That's cool.
Now, snakes don't have external ears. They do have internal ears, however. So they can hear sounds, but they can only hear sounds that are vibrating through their skull. So it's like if you plug your ears and you make sound, you can still hear it, right? Because it's vibrating through your skull. And your skull is vibrating that inner ear. So they do have cochlea, and they do have those hair cells, those organs of Corti, in the cochlea. So they can hear even though they don't have external and middle ear parts. It's the bones through the skin and through their jaw, because where's your jaw attached to your skull? Right here next to your ears. Right? So vibrations traveling through their jaw are transmitted by the bones in their jaw to their ear. And so that's why they'll sometimes put their jaw on the ground to listen for vibrations coming through the ground, okay, or through the air.
That's neat.
Now, we always got to go back to arthropods, right? The most
Chordontal. This is like a... Wait, cord? No. Cord...
successful terrestrial organisms on the planet, and they also have ears. They're not necessarily located in their head, because we said that. Now, we always got to go back to the arthropods, right? The most successful terrestrial organisms on the planet, and they also have ears, but they're not necessarily located in their head, because we said that they can make vibrations, they can make sound, so if they can use that to communicate, they have to be able to hear sound. And so the organ they use to hear is called the chordotonal organ. So the tone, sound, chords. So these sound chords.
Oh, chordotonal. I thought it was... Oh, I had a little dyslexic moment. I thought it was daunt. Yeah, see what I wrote? Chordontal? Chordotonal. Chordotonal.
So it's somewhat analogous to hair cells, only instead of bending the cilia, they're vibrating them. And so it looks something like this. So here's the neuron. Here's these dendrites sticking out here, and they're kind of strung like the strings on a guitar, or more accurately, like the strings in a piano. And so when something vibrates these things, they vibrate. And there's a little wide spot here with a crystal of chitin that gives a little more mass to it, right? And so you get this thing moving a little bit and it'll vibrate back and forth.
That's sick.
And so it's, again, bending this sensory cilia that's going to stimulate this neuron. So they're vibrating like the strings of a piano. Okay? And so these can be located in their antenna. They can
The
be located in
freaking
their legs, but they
resonator
can be
thing,
anywhere.
that's crazy.
These vibration sensors.
It's a little tuning fork.
Now, sometimes they're even more like an ear, where they actually have an eardrum. So
Bro, I hate that feature. Tympanum.
drum membrane over an airspace. So again, thinking of like a bass drum or bass guitar. So you have this like window here of this tympanic membrane, and the chordotonal organs will be underneath that. And so when things vibrate that big plate, it's vibrating those stereocilia underneath it. And so they can be located in different places. So this is places in different insects. It's not like one insect has all these different ears, but different insects can have these ears in different places in its body, either on its head, and its thorax, and its abdomen, and its legs. So in the case of the cricket, it's here in its forelegs, right? So they're rubbing their legs, making those sounds, listening for sounds in their forelegs. In the grasshopper, they're actually on the side of their body. You only see it in the adults because the adults are the only ones that are communicating because they're doing it to mate. Okay, so chordotonal organs then.
Wait,
the
we don't have
ears of
14?
terrestrial arthropods.
But presumably there's like another lecture on this, because, yeah, sensory system part 2, because there's no way that this is, and there's just so much material.
Again, remember they have that tracheal air system where they already have air spaces in their body, the tracheal air system.
Dude,
So
I
you have these air
actually,
sacs
this is like my
that,
favorite
again,
one
like
so
a
far.
bass
Is
guitar,
the
the
neurons
air
and
sac
sensing.
is going to amplify those vibrations.
Amazing.
So that's could be passive, or it could be active. So passive, you're listening to vibrations created by the environment, including by another individual. With active hearing, you're making the sound and then listening for how that sound is changed by the environment. So we often call that echolocation, right? You're listening to the echo of the sound that you made. So active hearing. So we see some degree of this in a variety of different animals. So in seals, for example, underwater, they've got these big bristles feeling for vibrations in the water, but they can also create sounds underwater and listen for those vibrations, because a lot of times these seals are down pitch black. They're so deep when they're diving. It's completely dark down there. And they can passively feel for vibrations in the environment. They also can make noises themselves and feel the echoes of their own vibrations. Oil birds are birds that that hang out in the daytime in caves, and when they head out at night to go feed, they have to be able to navigate the huge flocks of birds, not run into each other, not run into the walls of the caves. And they also will echolocate their way to not run into each other as they're migrating in and out of caves at night. Shrews are like living in the ground eating worms. I have no idea why shrews need echolocation, but apparently they do. So it's sometimes, we detect things, the presence of things, we have no idea what they're using them for. Or we may have ideas, but no way of testing those ideas, or at least we haven't tested them. Of course, if you want to look at really high, really refined echolocation, then we're talking about bats and whales. And so the extremely sophisticated, the ability to fine tune and resolve fine detail using echolocation. This is truly sonar. And so again, to locate prey, to navigate, to communicate, all of these things, they can create these vibrations and listen to their own vibrations or listen to others' vibrations. And interesting with a lot of these, particularly in the insectivore app, so not in the fruit bats, they don't do this, right? Because you're not trying to hunt fruit by echolocation. These are primarily in your predatory bats, particularly your insect-eating bats. And so they're able to locate insect-sized prey flying and moving around well enough to be able to consume large numbers of these insects. So they're incredibly efficient at even things as tiny as mosquitoes, catching them in the air and catching enough of them to feed themselves and their homeothermic little bodies. And so the frequency of the sound will often determine how precisely you can navigate. And so we see that when they're just navigating like through the caves or they're not moving into each other, they'll use a lower frequency sound. But when they're looking at high detail, like they're zoning in on a prey item, they'll make higher pitched, higher frequency sounds to be able to resolve more details, so higher acuity type signaling as opposed to perhaps higher sensitivity, lower resolution type echolocation. So they'll vary the type of location they do. And so there's all kinds of kind of arms race going on here between predator and prey at getting better at echolocating and getting better at avoiding being echolocated. So for example, we have this moth and this bat, where the bat is echolocating and catching a large number of these moths to feed on.
How?
But the moths, in turn, individuals are getting better at avoiding being located. And so some of the strategies that they use to try to avoid being caught by bats. And sometimes the bat doesn't have to catch you in its teeth, it'll actually catch you in its wings and then pass it from their wings into the mouth while in midair. So we talk about the bats making these sounds and echolocating on them. The moths have chordotonal organs, they have ears. And so they're not listening to sounds made by the moths, they're listening to sounds made by bats. And so here's a series of air sacs around those chordotonal organs. And here's that tympanic membrane. The tympanic membrane's here, air sacs to amplify the vibrations, the chordotonal organs to transduce those vibrations and nerve impulses. So they've got ears, listening for bat sounds. So the bat is active hearing, right? It's echolocating. It's making sounds and listening for its echoes. And it can detect something the size of a moth within about 3 meters. I guess that's a pretty good distance to detect something that small while you're flying. The moth is doing passive hearing. It's listening for sounds made by bats. Now, interestingly, a moth can't outfly a bat. So the strategy of the moth depends on what type, not only that it's detecting that there is a bat, but what type of frequency is the bat making. Because if it hears those low pulse rate sounds, that's the navigation sounds of the bat. And so then the moth will just change direction and try to fly away because the bat hasn't really detected it yet. It's just a bat is in the area. Whereas if it detects the high pulse rate, then it knows that the bat is locking in on it now. And so it won't try to outfly the bat, it'll just fold its wings and fall out of the sky. So it's like it not only detects the presence of the bat, but if the bat is locked in on it or not. And then if it doesn't, it tries to fly away. If it does, just fall from the sky and hope you fall faster than the bat can catch you. There's even one species of moth that stridulates, rubs its wings together in such a frequency that it jams the bat signal. So it's got sonar jamming capability.
Come on. Let's hear him say that one more time.
In such a frequency that it jams the bat signal. So it's got sonar jamming capability to it. It's amazing stuff going on.
I'm just going to write that down. Moth sonar jamming. That's so sick, dude.
Now, the other amazing echolocators are the toothed whales. So Odontocetes whales, odonto teeth. So the baleen whales.
I didn't know seats mean whale. That's why I'm writing it down.
Aren't doing this, and they do make sounds, but they're not doing echolocation. The toothed whales are. So the dolphins, sperm whales are doing this echolocation. And so in order to make the sounds, now again, when we make sounds, we're exhaling, right? Marine mammals don't want to lose the air. And remember, in many cases, marine mammals actually exhale before they dive. So they didn't want to be losing a lot of air making sounds. So they're not using their larynx to make the sound. They have a structure up here near their blowholes. The blowhole is their nostrils, but that's on the top of their head instead of on the front of their head. And there's a structure in here called the monkey lips. Monkey lips looks like these big fat lips together, and a couple of air sacs on either side of it. And so those air sacs squeeze the air back and forth through this monkey lips. So just that sounds. So just like you make sound go through a tight space, it's going to make vibrations. So it's generating the sounds up here at the monkey lips. And this air is passing back and forth. So it's not losing the air; it's passing it back and forth, creating vibrations. Now, as those sounds are being emitted out the skull of the animal, if we look at when we do our vertebrate lab, you'll see that dolphins have this really long rostrum on them. And that's because that's where this thing is sitting. In their forehead here is this big jello-filled thing called the melon. And that has a different density than the surrounding media. And when sound waves go from one media to another, they bend. So this is used as a lens to focus the sound. So they actually have muscles that can change the shape of this melon and focus that sound. So they're creating the sound here, focusing it through the melon, so they can pinpoint exactly where that sound is going.
Sure. Why
So
not?
in some cases, they use it actually to hunt that way. They'll actually stun the fish.
That's crazy.
By
A
bouncing
freaking
sounds
wave
off of it,
guide.
stunning the fish. But when they're echolocating, they're listening to the echo
A
of that sound
biological
coming back.
wave guide. Are you guys hearing
Now,
this?
how are they receiving? So that's how they're transmitting the signal. How are they receiving the signal?
Insane, dude.
Well, you don't want it coming back in the way it came out because that way, otherwise, the signal will interfere with itself. So instead, they receive the signal through their jaw.
Oh.
So their jaw has an oil-filled canal in it that focuses the sound down the jaw. And remember, like we talked about in the snakes, the ears are articulated with the skull where your ears are. So the ears are down here. So that way the sound comes out this way, comes back this way. It's not interfering with itself. And that allows them to do this really fine resolution where they can detect if you have something the same size but different material. It can detect a dense material from a less dense material, let alone the size and shape of things. We can give each other sonograms. And it's amazing the fine detail they can do with their ability to echolocate. And we see a similar sort of arms race like we did between the bats and the moth, between the dolphins and the fish. Because sound waves are going to bounce better off of something hollow than off of something solid. So if you have a fish with one of the swim bladders we talked about, then that airspace in the swim bladder is going to bounce a sound off. It's going to be really easy for a dolphin to detect a fish with a swim bladder. But certain fish have learned to detect that when a dolphin is bouncing sounds off its swim bladder, that a dolphin's honing in on me, it's time to start swimming erratically and make it harder to catch. So they've learned to listen to dolphin sounds and take evasive action. So
So there's antagonist, adversarial, coevolution.
So the better the dolphin is at locating the fish, the better the fish is at detecting that the dolphin is locating it. Okay, so that's echolocation. Now, some other types of waves that travel through the environment are things like other pressure waves that aren't necessarily those same kind of frequency. And overall pressure. Now, how do you detect pressure? That's a little tougher because it's not waves, it's more of a constant thing. And so it's not real well understood how animals are able to detect changes in the overall pressure of the environment around them. But again, we know that they do. So because we don't do it so much, we haven't really investigated it that much because we don't really think it must not be important because we don't do it, right? The whole anthropocentric thing. But we know that many animals are able to detect pressure changes in the environment around them. So in aquatic animals, for example, they can tell how deep they are. That one of the largest migration of animals on the Earth happens every day, where at nighttime, this huge mass of life travels up near the surface to a certain depth, not all the way to the surface, to a certain depth. And then during the day goes back down to a certain depth and stops. So it's not like it just keeps going up and keeps going down. It goes up to a certain depth and stops. How does it know how deep it is? Somehow it can feel the pressure. Or bats, when, again, you're in caves during the day, at night, they can actually detect the weather, right? You look at the weather report, they talk about low pressure systems, high pressure systems, telling you about storms. The bat's in its cave, it doesn't know if it's raining outside, but it can tell by the pressure, the air pressure, whether or not a storm is present. And therefore, if there's a storm blowing, no sense going out trying to echolocate and fly in a storm. Hang out. And so they only will go out when the conditions are right. That makes it likely that they're going to catch prey. How do they do that? There are mysteries yet to be solved. Now, again, as we've alluded to, water transmits pressure waves much more effectively than air does. And so vibrations underwater travel much more intensely than they do through air. And so, not too surprisingly, many aquatic animals are really good at detecting vibrations, even more so than terrestrial animals. And so this allows them to really feel their environment in ways that we really can't think of. It's like if we're moving through the room, right, we're displacing air and pushing. Air off the walls, we don't really feel that. A fish can feel that. A fish, as it moves through the environment, is displacing water. It can feel that water bouncing off its environment and feel the environment around it. It can perceive the world around it by these vibrations. It's amazing stuff. So whether you're locating prey, something swimming in such a way, not only is it present, but how is it swimming? Avoid predators, feel if something's coming at you, they're creating this pressure wave in front of them. You get schooling fish. You see this school of fish, and these thousands of fish, they're all moving in concert with each other, keeping this exact space between them, feeling where the other fish are around them. To feel currents, like which way is the ocean moving. The fish often will swim into the current or with the current. They have to be able to detect which way the current's coming from. And again, there's navigating your way through the kelp forest or through a coral reef. You have to be able to feel where the environment is and not just count on seeing it. In the dark or in murky water, you still need to not bump into things. So the pressures detecting system of fish is a series of structures along the sides of their body, arranged in this canal and these modified scales. These modified scales lined up along the sides of their body with these canals filled with this fluid, and then these sensory structures called neuromasts. So here's a blow up of the neuromast, and that should look familiar because that looks a whole lot like that inner ear structure. So they're able to have hair cells like we do in our inner ear. Well, instead of having one inner ear on either side, they've got a whole series of inner ears running the length of their body. So they're not only detecting sounds coming to their head, they can feel vibrations coming at them from any direction. So it's a lateral array running down both sides of the body. So this lateral line system allows them to feel the environments. These collection of hair cells into this kind of little jello capsule here called the cupula. That, again, you bend the hair cells one way, you increase rate of firing, bend the other way, you decrease the rate of firing. And their brain is able to process all this information. Now, in sharks, it's even better developed. It's an analogous system. It seems to be very similar, but yet embryologically derived differently. So how do you get the exact same system independently? It's like amazing stuff. But sharks don't have scales. So their canal is running under their skin, but with pores coming out to the surface, but still, again, these neuromasts arranged in the lateral line. And the lateral line not only runs the length of their body, but in their head, branches out around their face. So it's a branch of it around their eyes and around their mouth. So they can feel if something's coming at their eyes, which is vulnerable. They can also feel what they're biting at by its position in the water. They can feel where it is by the vibrations in the water. Okay, because their mouth is down here, right? They can't really see what they're biting, but they can feel what they're biting. Okay, so those are systems to detect vibrations and pressure waves in the environment. Now, another system that uses hair cells that's also in the inner ear is your vestibular apparatus. And we're not going to really talk about this.
Oh, shit, dude, I hate that. It's like so easy to freaking...
Fluid, orange cell, they can feel where it is by the vibrations in the water. Cells that's also in the inner ear is your vestibular apparatus. And we're not going to really talk about this extensively, other than that there are these utricle and saccule system here and these semicircular canals that also have hair cells in them. But they're detecting more your motion and orientation of gravity. So they're gravity sensors and motion detectors. Because it's an inertial system that if, for example, you have these little rocks in your head called otoliths, and so if they're sitting on the hair cells down here, you know gravity is pushing that way. If you turn your head, then now the rocks are over there and they're hitting the cells over here. So you can tell the difference of gravity. And then if you accelerate, then inertia, right, it's going to push the hair cells the other way. So if you hit this way, inertia pushes them back. You can tell acceleration. And so you can tell linear acceleration in the saccule system and then angular.
Bro, they do not have a freaking accelerometer. We all do? What?
Acceleration with these semicircular canals. The fluid sloshes through these, which are oriented in 3 different planes. So you can tell
That's what that is? Dude, not a fucking shot. That is so sick.
your motion in any arc as you whirl around. And so that's how your body knows where your head is and what direction you're moving. And then all those proprioceptors come in and say, okay, if my head's moving this way, the rest of my body is here relative to my head, therefore I know where my body is. So you look at a gymnast or a skater or an acrobat, and they do all these amazing things and they know exactly where their body is. It's this inner ear working with those proprioceptors and a brain doing a whole lot of processing of information. So we can use this sort of gravity thing that if you're sitting here, then those ear rocks are sitting on these hair cells. You move forward, and now the ear rocks are shuffling that way, you can tell which way gravity is, or linear acceleration. Whereas if you're rotating, then it's more the fluid in those semicircular canals. The fluid sloshes one way, so the fluid sloshes that way, it's going to bend the hair cells that way. It's all inertia. So using inertia to detect acceleration and changes in velocity. And so we see differences in animals that need to have really good control of their balance, tend to have really big semicircular canals and vestibular systems here. So you get something arboreal like this brush baby that's jumping from branch to branch in the middle of the night. They need to have really good balance. So proportional to their body, they've got these huge semicircular canals compared to us. Whereas something like a dolphin, you know, dolphins, they're buoyed up in water. They don't have to worry about falling. They're little tiny semicircular canals.
Wow.
And again, we see in
Dude, I cannot. Something's with my mouse.
the same thing that our utricle and saccule system are doing. And anatomically similar, even though derived completely independently. And just a little sidebar that there are the gravity sensors in plants, gravity sensors in fungi, that it's not just animals that have sensory systems, that all living things respond to changes in the environment. And so here, for example, in this fungus, there are these, you know, it needs to put its sporangia up in the air to disperse its spores. Well, how do you know where up is? You have to have gravity sensors. So it's similar to those statoliths. You got a little sphere with a little crystal in it. And so you can tell by where that crystal is in the sphere which direction up is. Okay, so and we talked about in plants, you know, the shoot system grows up, the root system goes down. How does it know up from down? They have to have gravity sensors of some sort. Okay, so those are the basics of how physiological transducers work. And applying it now, so we talked about chemoreceptors, we talked about mechanoreceptors. Primarily those are the two we talked about, in a variety of different organ systems. Now we get to electromagnetic systems, the mechanisms are a little bit different. So I think we'll take a pause here. And we'll come back in our next lecture and pick up with electromagnetic receptors,
Nice.
particularly vision and light, and other types of electromagnetic receptors.
Fantastic.
And we'll pick that up in our
Good,
next
great
lecture.
stopping point. All right. Peace.